Hello everyone and welcome to our channel MooTechie. A warm welcome to all the dairy technologists and students joining us today. We're diving into the fascinating world of fermentation technology with a special focus on probiotics. Moving on, we will see the key areas where fermentation technology makes an impact. These include improving food security, increasing income and employment, providing health benefits, enhancing cultural and social well-being, and understanding malolactic fermentation. Now, let's explore how fermentation improves food security. This technology plays a vital role in ensuring food security worldwide. It helps through improved food preservation, salvaging waste foods, and removing anti-nutritional factors. For example, in food preservation, fermentation extends the shelf life of foods. It also allows us to salvage waste products by fermenting them into edible food items. Additionally, it removes anti-nutritional factors. For instance, in kawai fermentation, toxins are removed from leaves. Fermentation also involves antimicrobial activities. Lactic acid bacteria produce organic acids like lactic acid, formic acid, propionic acid, and acetic acid, which lower the pH and inhibit undesirable microorganisms. They produce bacteriocins, which are bactericidal polypeptides or proteins that are heat stable at acid pH. Hydrogen peroxide is another antimicrobial. It is bacteriostatic at low concentrations and bactericidal at high concentrations. Diacetyl ACE, produced by several species of lactic acid bacteria through citrate metabolism, also has antimicrobial effects. Ethanol, produced during fermentation, is inhibitory to other microorganisms. Next, we define bacteriocins. Bacteriocins are ribosomally synthesized single polypeptides or post-translationally modified ones that are usually inhibitory only to closely related bacterial species. Let's look at a table categorizing bacteriocins from lactic acid bacteria. The main categories are Class 1, 2, 3, and IV. Class 1 are lanthiotics with subcategories Type A and Type B. Examples include niacin for Type A and mersacidin for Type B. Class 2 are non-modified heat-stable bacteriocins with peptides under 10 kilodaltons. Subclass Titia examples include pediocin ACH, enterocin FH99, and pediocin 34. Other Class 2 examples include lactocin F and lactococcin G. Class 3 are protein bacteriocins with masses around 30 kDa, such as helveticin J and lactocins A and B. Class 4 are bacteriocins that form large complexes. Now, we examine hydrogen peroxide in more detail. Hydrogen peroxide has a strong oxidizing effect on the bacterial cell wall. It can oxidize sulfhydryl groups of cell proteins and membrane lipids. Reactions that produce hydrogen peroxide also scavenge oxygen gas, creating an anaerobic environment unfavorable for some microorganisms. Its antimicrobial activity is enhanced by the presence of lactoperoxidase and thiocyanate. The chemical reaction involves thiocyanate and hydrogen peroxide forming hypothiocyanate and water. Following this, we have a table of some useful bacteriocins for the dairy and food industry. It lists the genus of bacteria and the bacteriocin they produce. For example, Lactococcus lactis subsp. Lactis produces nisin A and Z. Pediococcus acidilactici produces pediocin PA1 and ACH. Pediococcus pentosaceus produces pediocin 34. Leuconostoc species produce leucocins. Lactobacillus ceacae produces sakacin A. Lactobacillus plantarum produces plantaricin. Lactobacillus helveticus produces helveticin J. Carnobacterium piscicola produces carnosin or piscicolin. Now let's discuss diacetyl. Diacetyl was identified by Van Niel and others in 1929 as the aroma and flavor component in butter. It is produced by species and strains of genera like Lactobacillus, Leuconostoc, Pediococcus, and Streptococcus. Diacetyl is more active against gram-negative bacteria, yeasts, and molds than against gram-positive bacteria. It works by reacting with the arginine-binding protein of gram-negative bacteria, interfering with their utilization of arginine. Moving on to health benefits, we start with improving nutrition. Lactic fermentation improves nutritional value by increasing levels of amino acids. It also breaks down large molecular-sized nutrients, increasing digestibility and reducing toxic metabolites. Next, vitamins. Saccharomyces cerevisiae can concentrate large quantities of thiamine, nicotinic acid, and biotin, forming enriched products. For example, palm wine in West Africa is high in vitamin B12, which is important for people with low meat intake. Then digestibility. Microorganisms produce certain enzymes not synthesized by humans, such as cellulases, which aid in digestion. Now, what are probiotics? Probiotics are defined by the WHO in 2002 as live microbes that, when administered in adequate amounts, confer a beneficial health effect on the host. A table lists common probiotic species. For Lactobacillus species we have L. acidophilus, L. casei, rhamnosus, L. reuteri, L. bulgaricus, L. plantarum, and L. johnsonii. For Bifidobacterium species, Mediv, we have B. bifidum, B. longum, B. breve, B. infantis, B. lactis, and B. adolescens. Other probiotics include Bacillus cereus, non-pathogenic Escherichia coli, Saccharomyces cerevisiae, Enterococcus faecalis, and Streptococcus thermophilus. Let's review the history of probiotics. Elie Mechnikov was the first scientist to propose the therapeutic use of lactic acid bacteria. Lactic acid bacteria were first discovered by Pasteur in 1857. Their isolation from rancid milk was reported by Lister in 1878. The first stable cultures of Lactobacillus casei strain Shirota were made in 1930 by Dr. Minoru Shirota. In 1889, Tissier discovered Bifidobacterium species. Why are probiotics important for human health? Factors like antibiotics, alcohol consumption, and stress can destroy the gut microflora. This leads to the emergence of antibiotic-resistant strains, problems in digestion, and issues with nutrient absorption. Probiotics help counteract these effects. Now, let's describe Lactobacillus and Bifidobacterium strains. Lactobacillus strains are acid-tolerant and produce lactic acid as a major metabolic end product. Bifidobacterium are saccharolytic organisms that produce acetic and lactic acids without generating CO_2. They are also considered lactic acid bacteria. Their sources include raw and fermented dairy products, fresh and fermented plant products, and the reproductive and intestinal tracts of humans and other animals. Next, we look at the features of probiotics. They should be non-pathogenic, non-toxic, resistant to gastric acid, high Z, and able to adhere to gut epithelial tissue. They should produce antibacterial substances, persist for short periods in the gastrointestinal tract, resist antibacterial mechanisms in the gut, avoid the effects of peristalsis, and be resistant to bile acids. The mechanism of action of probiotics involves producing low molecular weight antibacterial substances that inhibit both gram-positive and gram-negative enteric bacteria. They use enzymatic mechanisms to modify toxin receptors and block toxin-mediated pathology. They also prevent colonization of pathogenic microorganisms by competitive inhibition for microbial adhesion sites. A diagram summarizes the benefits of probiotics. These include suppression of endogenous pathogens like antibiotic-associated diarrhea, colonization resistance, suppression of exogenous pathogens like traveler's diarrhea, supply of short-chain fatty acids and vitamins to the colonic epithelium, reduction in risk factors for colon cancer, and lower levels of toxigenic or mutagenic reactions in the gut. Probiotics also normalize intestinal microbiota composition (SPECE), balance immune response (SPECE), provide immunomodulation (SPECE), strengthen innate immunity, lower serum cholesterol (SPECE), assist in bile salt deconjugation and secretion (SPECE), hydrolyze lactose boscos (SPECE), and improve lactose tolerance. Now, let's explain cholesterol assimilation by probiotics. The mechanisms are direct and indirect. Direct mechanisms include assimilation, binding, and degradation of cholesterol. Indirect mechanisms involve inhibiting the de novo synthesis of cholesterol, decreasing intestinal absorption of dietary cholesterol, and deconjugating cholesterol to bile salts. Compounds like lactose, calcium hydroxyl methyl glutarate, uric acid, and whey proteins play roles in this process. On anti-cancer effects, a high animal fat and protein diet can increase susceptibility to colon cancer by converting pre-carcinogens to carcinogens through intestinal microflora enzymes like glycosidase, beta-glucuronidase, azoreductase, azoreductase, and nitroreductase. Supplementation with L. acidophilus and L. casei in humans has been shown to decrease levels of these enzymes, reducing cancer risk. For lactose intolerance, normally lactose is digested in the small intestine. In lactose-intolerant individuals, yogurt is tolerated due to the lactase activity from lactic acid bacteria in the yogurt. Lactose deficiency can lead to calcium malabsorption, but the acidic condition from lactic acid production enhances calcium absorption. Probiotics help by hydrolyzing milk lactose. A table lists probiotics and their effects on allergies. For example, Lactobacillus rhamnosus GG helps with asthma, rhinitis, eczema, and food allergy. Bifidobacterium lactis helps with atopic eczema. Lactobacillus paracasei helps with allergic rhinitis. Lactobacillus reuteri helps with atopic dermatitis. Probiotics work by degrading or structurally modifying antigens, normalizing aberrant indigenous microbiota, regulating the secretion of inflammatory mediators, stimulating immune response, reducing serum IgE levels, and improving mucosal barrier function. They have been shown to reduce childhood eczema and control lactose intolerance. Another table shows commercial probiotic strains and their products. For instance, L. acidophilus is used in LA5 and NCDO 1748. Saccharomyces cerevisiae, boulardii, is in Florastor. L. casei-shirota is in Yakult. B. Breve strain Yakult is also used. L. johnsonii LJ1 is in LC1 by Nestlé. L. reuteri ATCC 55730 is in BioGaia products. Streptococcus oralis KJ3 and Streptococcus rattus JH145 are in Probiora 3. Lactobacillus thermosus PB01 and Lactobacillus gasseri EB01 are in Ecovag. Moving on, fermentation improves cultural and social well-being by enhancing the flavor and appearance of foods. For example, furundu, a meat substitute, is made by cooking carcade seeds and fermenting them for 9 days. Fermented vegetables are used as condiments to enhance meal flavors. Gundruk is a fermented green leaf product, often made from leaves like radish or mustard. Now let's discuss malolactic fermentation. This process, also known as malolactic conversion or MLF, is used in winemaking. It converts tart-tasting malic acid in grape must to softer-tasting lactic acid. Lactic acid bacteria decarboxylate L-malic acid to produce L-lactate. This fermentation provides bacteriological stability to bottled wine and can improve its body and flavor. There are many main fermented foods produced around the world. These include milk-based products like kurut, kefir, and kumis, cereal-based products like tarhana, boza, mahevu, idli, and dosa, meat-based products like sucuk and pastirma, fish-based products like fish sauce, soy-based products like soy sauce, natto, and tempeh, and vegetable and fruit-based products like kimchi, sauerkraut, gundruk, and sunki. Let's describe mahevu. Mahevu is a traditional Southern African non-alcoholic drink made from maize meal. While home production is common, it is also factory-produced and available in supermarkets. Next, idli. Idli is a very popular fermented breakfast food in the Indian subcontinent, made mainly from rice and black gram. The process involves fermenting a batter of rice and black gram. Then dosa. Dosa is a rice-based crepe made from a fermented batter of rice and black lentil. It is highly popular and consumed as a breakfast food. Now tempeh. Tempeh is a traditional soy product from Indonesia. It is made by a natural culturing and controlled fermentation process that binds soybeans into a cake. Finally, sunki. Sunki is a non-salted fermented vegetable product from Japan, made from the leaves of the otaki turnip. The leaves are boiled, inoculated with dried sunki from the previous year using zumi, a wild apple, and fermented for 1 to 2 months in low temperatures during winter. Thank you for listening to this comprehensive session on fermentation technology and probiotics. We've covered how fermentation enhances food security, the role of antimicrobials like bacteriocins, the health benefits of probiotics, their mechanisms, and examples of fermented foods worldwide. Understanding these concepts is key for applications in dairy technology and related fields.